Today we're going to go over the answers to the Punnet square problems that are in the study guide. So number 1 says that tall eyeballs are dominant to short eyeballs. Mr. Crab and Mrs. Crab are both heterozygous for their tall eyeballs. If you recall, a heterozygous genotype means that there's two different alleles for that gene. And that means that the genotype for Mr. Crab is going to be capital T, lowercase t, because that's a heterozygous genotype. And since they're both heterozygous, Mrs. Crab will also be capital T, lowercase t. The next thing we need to do is create a Punnet square that shows this cross. So you're going to put one of the parents on the top of the box, the other parent on the side of the box. And then if you recall how to fill in the boxes, it should look like this. And from that information, we can identify the answers to the next two questions. So the first question says, what are the chances of them having a child with tall eyeballs? Recall that tall eyeballs can be caused by two different genotypes. So somebody with tall eyeballs can either be capital T, so that they're homozygous for tall eyeballs, or like Mr. Crab and Mrs. Crab, they can be capital T, lowercase t, and be heterozygous and still show the dominant trait. So we know this is going to be out of 4 because there's 4 different possibilities. And when we look at this, what you should notice is that there are 3 options that give you tall eyeballs. Remember, those 3 options are the homozygous dominant and the 2 heterozygous dominant genotypes. That means that there's a 75% chance that their offspring will have tall eyeballs. The next question says, what are the chances of them having a child with short eyeballs? Remember that short eyeballs are the recessive trait. And so in order for something to have short eyeballs, it has to have 2 copies of the recessive allele. So the genotype would be lowercase t. And so here we're looking for just this individual, lowercase t. That means there's only 1 out of the 4 offspring that have a chance of having short eyeballs. And that means it's 25%. Number 2 says that brown eyes are dominant to blue eyes. A man is heterozygous for brown eyes, but his wife has blue eyes. This doesn't tell us what letter to use, and so we're going to use the letter capital B for brown eyes and the letter lowercase b for blue eyes. And then we can figure out our genotypes. It says that the man is heterozygous, so that means that his genotype is going to be capital B, small b, and his wife has blue eyes. Remember that blue eyes is recessive, so her genotype is going to be lowercase b. Then we can put these 2 individuals in a Punnet square, and then we can figure out the probability of their children having either brown eyes or blue eyes. So it says, what are the chances of a child with brown eyes? Since brown is dominant, they have to either be capital B, or capital B, small b. And you'll notice that 2 of their potential offspring have brown eyes. So that's 2 out of 4. That's the same thing as 50%. So there's a 50% chance their child will have brown eyes. And then this next question says, what are the chances of a child with blue eyes? So blue eyes can only happen if you have small b as your genotype. So this means that 2 out of 4, or 50% of their children have a chance of having blue eyes. The next problem that we're going to go over is incomplete dominance. So the question says that SpongeBob loves growing flowers for his pal Sandy. Her favorite flowers, pumpkins, show incomplete dominance that are found in red, blue, and purple. R is the red gene and B is the blue gene. So in order for something to be red, it must have the genotype capital R. In order for something to be blue, it has to have the gene capital B. And the genotype for purple is 1 red allele and 1 blue allele. So the question says that SpongeBob crosses a purple pumpkin with a blue pumpkin. So we need to put the purple genotype on one side of our Punnet square and the blue genotype on the other side of our Punnett square. Then we can fill out our Punnett square, and that's going to tell us the probability of what color flowers are going to result. So the question here says, how many of the plants would have red flowers? Remember that in order for something to have red flowers, it has to have the genotype capital R, capital R. Since none of the plants in our Punnett square are capital R, 0% of the offspring will have red flowers. The next question says, how many of the plants would have purple flowers? Remember, purple flowers are controlled by the allele R and B. So anytime you see R plus B together, you're going to have purple flowers. And since there's 2 out of 4, that is the same thing as 50%. The last question says, how many of the plants would have blue flowers? Remember that for something to be blue, you need the genotype capital B, capital B. So in this case, we have 2 plants out of 4, or 50%, that will have blue flowers. The next problem, number 4, involves both codominance and multiple alleles. So it says a father has AB positive blood and a mother has AB negative blood. And then we're going to answer 2 questions: whether it's possible for them to have a child with type O blood, and if they can have a child with B negative blood. So first, let's make a Punnett square for their type AB blood. Both parents have type AB, so we can start with that and complete our Punnett square. And then we can look to see if type O is a possibility. Remember that type O blood has to have the genotype OO. So none of these children possibilities have type O blood. That means that the answer to our first question is no. They cannot have a child with type O blood. Next, we answer the question, is it possible for them to have a child with type B negative blood? We know that type B blood is a possibility because the genotype capital B gives us type B blood. And we also know that one of the parents has the allele for the Rh negative factor. Even though this is recessive, if the father is heterozygous, the child could have type B blood. So the answer is yes, if the father is heterozygous. Now let's move on to number 5. It says, in each case below, one parent has type A blood and the other has type AB blood. So we know their blood types, we just need to figure out their genotypes. So we're going to figure that out by working backwards from what their children look like. So in case A, half of their offspring have type AB blood and half of their offspring have type A blood. Because none of the children have type B blood in this instance, the likely genotypes for this are AB and AA, and we can confirm that by completing a Punnett square. So let's put AB on the top of our Punnett square and then AA on the side and see if this gives us 50% AB and 50% type A. And you'll notice that is in fact what happens. So you can see that half of the children have type A blood, which is what we were looking for. The other half of the children have type AB blood, which is the other condition we were looking for. So our answers to this question for parent 1 and 2 genotypes are AB and AA. In the second case, we're looking for parents that have offspring with the following genotypes. They should have half A, a quarter AB, and a quarter B. We know that one of the parents' genotypes is AB because that was told to us at the beginning of the problem. And so we're going to put that on one side of our Punnett square. And then the other parent has type A blood. We know that it's not AA because we just made that Punnett square and it does not give us any type B offspring. So the only other way to have type A blood is to have the genotype AO. And so this is most likely the genotype of our parents, but let's confirm by completing the Punnett square. And now you should notice from the Punnett square that we do meet all of these conditions. Half of the offspring are supposed to have type A blood, and that is what we see here, 2 children that have type A. One is homozygous for the A alleles, one is heterozygous but still type A. Then we're looking for 1/4 of the children to have AB, and that is true. We have one person who has type AB blood, so that's that one. And then we're looking for 1/4 of the children to have type B blood, which is here with the genotype BO. So indeed the parents in this particular Punnett square should be an AB parent and an AO parent. Next we're going to look at a sex-linked trait or X-linked trait. So hemophilia is a sex-linked recessive trait with the gene located only on the X chromosome. We're going to use XH as the gene for normal blood clotting, and X_h is the gene for hemophilia. So we're going to indicate phenotypes and ratios for the following cross between 2 different people. So the mother is heterozygous, and that means that she would have the genotype XH, X_h. So she carries one gene for hemophilia and one gene that is normal clotting factor. This means that she is a carrier. We also need the genotype of the father. So he has hemophilia, which is what we learned here. So he's going to be X_h, and then his other sex chromosome is a Y chromosome. And this means he has hemophilia because he has the trait. So now we're going to look at the chances for having the following children: a normal female, a female with hemophilia, a normal male, and a male with hemophilia. So to make our Punnett square, we can do XH, X_h on one side of our Punnett square. We can do X_h and Y on the side of our Punnett square. And then we'll fill out our Punnett square. And we have one box that has XH, X_h. We have one box with X_h, X_h. And then we have one box that's XH with a Y, and then X_h with a Y. What this is going to give us is our results. So first we'll look for a normal female, meaning a female who has normal clotting factor. This includes females that are carriers. So this individual, XH, is going to be a normal female who can clot. That's 1 out of 4 means there's 25% chance they will have a female who can clot as their child. We're also then going to look for a female with hemophilia. The genotype for that is X_h, X_h. So this female has hemophilia. There's one of them, and so that's also 25%. Next, we're going to look for a male who has normal clotting factor. And so there is indeed one option, XHY, and since there's only one, there's a 25% chance that they'll have a male who can clot. And they also do have a chance of having a male with hemophilia. X_hy is the genotype that would give us that, and so that's again 25% chance since it's 1 out of 4. In our last problem, we're going to use 2 different traits, and we're going to look at a dihybrid Punnett square. Both traits have a Mendelian inheritance pattern. So first let's look at our traits. It says that in some flowers, tall is dominant to short, and orange petals are dominant to white petals. So those are the 2 traits we're going to look at. We're going to use the letter T for height and A for petals. And then we're going to complete a cross where both parents are heterozygous for both traits. So because both parents are heterozygous for both traits, they're going to have the same genotype. The genotype for 2 heterozygous traits would be capital T, small t, because they're heterozygous for height, and then capital A, small a, because they're heterozygous for petal color. And since the parents are identical in these 2 traits, their genotypes for these 2 traits will also be the same. The next thing that we're going to do is look at how to generate the gametes. Remember, a gamete is a sex cell, and it has half the amount of DNA that a diploid or a parent cell does. And so those gametes are going to have 2 letters instead of 4. So the gametes that are generated by the genotype capital T, capital A, small a are capital T, capital A, right? capital T, small a, because that's another way to combine the letter T with the letter A. Small t, capital A. And the last possible combination is small t, small a. So those letters are the ones that are going to go on our Punnet square. So next you can set up your Punnet square. You should notice that the letters are the same on the top and the side of the Punnet square, and that's because the parents have the same genotype for these two traits. Next, you're going to fill in your Punnet square so that each box has 4 letters. If you need to pause the video for a moment to write down the Punnet square, feel free to do so. Next, we'll answer the last 2 questions. The first question says, how many plants will be heterozygous for both traits? So here we're looking for the genotype, big T, small t, big A, small a. And you should find that there of the 16 that are there, there's 4. And so you're looking for these genotypes, right? Big T, big A, small a, and there's 4 of them. So 4 out of 16. The next question says, how many plants will be short and have white petals? So the genotype for short is small t, and the genotype for white petals is small a, small a. So you're looking for this genotype in your Punnet square, and you should find that there is only one individual that has small t, small a, small t, small a, small a as their genotype. So 1 in 16 of these individuals will be short and have white petals.